And then I have one final task here, and that is to I know some students like images, and then some students need a sequence of events or something that's more written. So I've tried to kind of accommodate all learning types here, or learning preferences. And in this one, I have all the steps. And what you just need to do is rearrange these into the correct sequence of events that happens. So you can go ahead and pause and then move these around. They aren't post-its, but you can move the texts around. So see if you can get these in order, and I'll come back and we'll double check to see if you got them. So now some of these you may have in a slightly different order, but first thing that needs to happen is that the DNA has to open, and it opens specifically at that origin of replication. So now it's open, and then now we need to get it started. And in order to get it started, we need to unwind it further, right? So that helicase is going to unwind the DNA, and then, handily enough, the next one's already here, where that primase needs to bind, and we need to get the RNA primer. So we opened it up, and now we have room for the primase to come in and synthesize an RNA primer. And then these two could, again, the next two that could come into play. So part of it is going to be single-stranded, and so those single-stranded binding proteins need to come help and stabilize that single-stranded DNA. But then also at the same time, we have to have that topoisomerase, because now we're opening it up with the helicase. It's going to function ahead of the replication fork to break and swivel the DNA so that we don't have breaking of our DNA. And then we are going to now add our nucleotides. And the enzyme that does that is that DNA polymerase 3, again at the 3' end of the primer, and going to add those DNA nucleotides. And then we need to, once that's all done, right, we need to get rid of the RNA primers. So then DNA polymerase 1 will come in and get rid of those RNA primers, and then DNA ligase is going to come in and finish it and seal off any missing phosphodiester bonds that we have. So that is all of the components. That's all the steps of that DNA replication that is happening during S phase in order to make copies of cells.